Have you ever wondered what happens when you smash biology and computer science together? Well, today we're diving into bioinformatics, the field that pretty much became the unsung hero of the COVID-19 pandemic.
So let's start with the big question, right? Figuring out a new disease used to take, well, decades. But with SARS-CoV-2, we went from identifying the virus to developing a vaccine in less than a year. How on earth did that happen? Well, the answer wasn't just found under a microscope. A huge part of it was on servers, inside algorithms, all thanks to the power of bioinformatics.
To really get a handle on that incredible speed, we first have to understand the tool itself. So, let's break down what bioinformatics actually is and why it's such an absolute game changer for biology.
Okay, so at its heart, bioinformatics is basically the bridge between biology and big data. Modern biology generates just mountains of raw data. Think DNA sequences. And bioinformatics gives us the tools to make sense of it all. Here's an easy way to think about it. If a genome is a library with billions of books, bioinformatics is the search engine that lets you find the exact sentence you're looking for instantly.
And this really shows you the difference. Traditional biology happens in vitro, in a test tube or a petri dish. It's slow, it's manual. Bioinformatics, on the other hand, happens in silico, on a computer. It is incredibly fast and automated, and it can turn what would have been months of painstaking lab work into just a few hours of computation.
All right, let's put this into practice with our main case file, the COVID-19 pandemic. Imagine we're detectives, and our mission is to track this invisible enemy, the SARS-CoV-2 virus. And bioinformatics, that's our #1 investigative tool.
This was the question when variants like Alpha, Delta, and Omicron started popping up. The virus was changing, and some of these new versions were spreading like wildfire. But why? The clue, it turns out, was hidden deep inside its genetic sequence, in the part that codes for one very, very important protein.
And this is the core mechanism right here. The virus has this tool, the spike protein. You can think of it as a master key. And to infect us, that key has to fit perfectly into a specific lock on the surface of our own cells, a receptor called ACE2. The better the fit between the key and the lock, the easier it is for the virus to just waltz right in and start making copies of itself.
So how did scientists figure this out so fast? Well, they used bioinformatics to run these incredibly complex computer simulations. They looked at a specific mutation called N501Y, the one in the alpha variant, and found that it strengthens the electrostatic interaction. Now, that's just a fancy way of saying it created a stronger, almost magnetic pull between the virus's key and our cell's lock. It made the key stickier, which made the virus way more infectious.
So if you're one of these genomic detectives trying to track a virus in real time, what's in your toolkit? It's not a magnifying glass and a notepad, that's for sure. It's a whole suite of powerful, connected databases and visualization platforms.
Just let this number sink in for a second. Over 6 million complete genetic blueprints of the virus were sequenced and uploaded for the whole world to see in just the first year of the pandemic. This was totally unprecedented. It created a real-time map of the virus's evolution that any researcher, anywhere, could access. And these were the command centers for making sense of it all. GSide was like the central library, holding all the data. NextStrain took that data and turned it into these amazing visualizations that track the virus's spread almost like a weather map. And then you had tools like covariance and outbreak.info that let scientists zoom right in on the specific variants they were most worried about.
Okay, so tracking a pandemic as it happens is amazing. But what's next? What's the next frontier? Well, it's moving from just reacting to actually predicting. And to do that, we're adding an incredibly powerful new tool to the bioinformatician's arsenal: artificial intelligence.
Yeah, this is where AI and machine learning become like a superpower. They can analyze millions of viral genomes and spot these incredibly complex patterns of mutations that a human researcher could just never see. It allows us to shift our focus from tracking what the virus has already done to predicting what it might do next.
So how does this actually work in practice? Let's walk through it. First, the AI just drinks up all that global genomic data from places like GSide. Second, its algorithms sift through all that noise to find subtle emerging patterns. Third, and this is the real game changer, it can actually start to predict which new combinations of mutations could lead to a really high-risk variant. And finally, this isn't just for a research paper. This foresight is used to inform real-world public health decisions.
And listen, this isn't science fiction. These kinds of early warning systems like IRAT and VAREPS are already being built and used. They're designed to evaluate the risk of new variants, figure out how bad the disease they cause might be, and basically help us get prepared before they become a global problem.
Which brings us to our last big thought here. We've seen how bioinformatics let us decode and fight a pandemic with incredible speed. But now, with AI joining the team, are we getting to a point where we can not only respond to outbreaks, but actually predict them and maybe even prevent them from ever starting? That is the future that bioinformatics is building right now.